The microbiome plays an important role in our health. We know that we all carry a huge number of species with us, and we know that we can change those species as a potential way of curing disease. But what we don't understand is how the individual bacteria and other microbes interact with each other. And that's what this research has really enabled us to do. So what we can do is we can understand how each of these bacteria behave as individuals and in combination, so in the communities. And by doing that, we can predict how they benefit each other, how they can compete with each other, and what the overall impact that will have on our health actually can be. What we really wanted to understand with this research was how the individual bacteria interact with each other, and we could predict this using computational models. In doing that, we could find the species that liked to live together, the species that didn't like to live together, and we could build up the communities in such a way that we could understand when they would be beneficial for health and when they might cause disease. In Crohn's disease, for example, we saw that there was a loss of a particular community state that was able to use up a detrimental chemical. By losing those species, you got an increase in the amount of the chemical, and that could drive inflammation and make the disease worse. It gives us an opportunity to intervene, changing the community structure, changing the individual bacteria that are present within that community, and shifting the community such that it doesn't drive the disease that we're trying to avoid. The microbiome represents a huge opportunity to treat disease. We know that changing the microbiome through fecal transplant can provide cures to people who are otherwise suffering from untreatable conditions. But what we don't understand is how the individual bacterial species work together, forming communities and providing that benefit. What this work allows us to do is understand those specific interactions, understand how the community of microbes will be operating within the person with a disease, and then intervene with particular bacterial species that will shift the community and, more importantly, the ability of that community to provide certain functions.